Hello. Module 11 starts with digestion and the transport of dietary lipids. This is the first session of a two-session module, and for this we'll look at Chapter 32. So there's two main focus areas of this session, the first being lipid digestion. Triglycerides are the major fat source in the human diet, and they're digested only marginally by lingual and gastric lipases. The majority of the digestion of triglycerides occurs in the intestine, and that's through the activity of pancreatic lipase. And the free fatty acids and monoglycerols are then packaged for transport. The transport of fatty acids and dietary triglycerides is via chylomicrons. Chylomicrons are able to transport otherwise non-soluble lipids into particles for delivery into the peripheral tissues. Chylomicrons have associated apoproteins, ApoC2 and ApoC48, which are essential for the function of these lipoproteins. So if we look at the overall schematic of digestion, again, it starts with an intake of dietary triglycerols. Digestion starts in both the mouth and the stomach through the actions of lingual and gastric lipase. Once the stomach contents empty into the small intestine, the triglycerols are emulsified by the addition of bile salts. From there you have digestion with both pancreatic lipase and chololipase into free fatty acids and monoglycerol, which are dissolved into micelles that facilitate the movement of free fatty acids and 2-monoglycerols into the intestinal epithelium. So again, this process starts with triglycerides. Triglycerides are molecules composed of a glycerol unit, shown here in green, and 3 fatty acid chains attached to one of each of the carbons. So this is our starting point for digestion. Small and medium chain fatty acids can be digested to a small extent in the mouth and the stomach, and longer chain fatty acids require the action of pancreatic lipase in the small intestine. So an important process as the dietary fats leave the stomach and enter the small intestine is the addition of bile salts. Bile salts are amphipathic compounds synthesized by the liver, and they're synthesized from cholesterol. The bile salts act as detergents and bind the globs of dietary fat, and this facilitates the breaking up of these molecules through the peristaltic action of the intestinal muscle. Emulsification of the fat increases the surface area of the compound, making it easier to be accessed by digestive enzymes. These digestive enzymes, chololipase and lipase, are both secreted from the pancreas. Again, pancreatic lipase and cholipase are secreted from the pancreas, and they're secreted in response to a hormone, cholecystokinin. At the same time, another hormone, secretin, signals the release of bicarbonate, and this increases the intestinal pH to about 6, which is a pH that is more amenable to enzyme activity. So initially, the coating of the bile salts around the triglycerides inhibits the pancreatic lipase. And colipase, which is released at the same time as pancreatic lipase, binds to dietary fats and relieves the inhibition of the bile salts. This enhances the ability of pancreatic lipase to hydrolyze triglycerides into fatty acids and a monoglycerol. This hydrolysis is shown here, where the initial triacylglycerol, with this being our glyceride, and our 3 fatty acid chains, are removed starting at carbon 3 to a diacylglycerol. This is followed by removal of the fatty acid at the carbon 1 position, leaving us with 2 monoacylglycerol. From here, these free fatty acids and monoacylglycerols can be packaged into micelles, which will contain both dietary cholesterol, phospholipids, free fatty acids, and monoacylglycerols. These micelles are tiny microdroplets that are emulsified by bile salts. The micelles will travel through a layer of water in an unstirred interface near the epithelium of the gut and will facilitate the transfer of the free fatty acids and 2 monoglycerols into the cell. The bile salts themselves are reabsorbed. More than 95% of bile salts are recirculated, and they travel through what is referred to as the enterohepatic circulation, and they're returned to the liver and are redeposited back into the gallbladder. In this manner, bile salts can be recycled through the digestive process. So if we look at that interface, again, we're talking about this brush border interface where our free fatty acids and our phospholipids and 2 monoglycerols are dissolved into micelle particles that can then interact with the brush border of the intestinal cells. From there, our lipid molecules can diffuse into the cellular space where they are repackaged into triglycerides for transport in our chylomicrons. So again, once the micelles interact with our intestinal epithelial cells, the contents diffuse into the cells, and again, our bile salts are recycled back to the liver and stored in the gallbladder. The free fatty acids are repackaged into triglycerides in the following scheme, where our free fatty acids are activated to fatty acyl-CoA here, and again, our 2 monoacylglycerol are condensed with our fatty acyl-CoA to generate a diglyceride and a subsequent triglyceride molecule. This repackaging of our fats and monoacylglycerols takes place in the smooth endoplasmic reticulum in the intestinal epithelial cells. This leaves us with our triacylglycerol molecule that will be transported by our nascent chylomicrons. These particles are required for the transport of triglycerols as they're insoluble in water. And if they entered the body directly, they would coalesce, similarly to the way that oil floats on top of water, and this would negatively impact blood flow. So a chylomicron is a lipoprotein that is comprised of triacylglycerols, cholesterol, and phospholipids, as well as apoproteins that are required for its function. This is a schematic of a lipoprotein, and here we're showing a chylomicron. Apoprotein B48, or ApoB48, is an apoprotein unique to chylomicrons. It involves a core of triacylglycerides, a monolayer of amphipathic lipids. Note that this is a monolayer as opposed to a cell membrane, which is a bilipid layer. It also incorporates cholesterol into this monolayer, as well as cholesterol ester. A cholesterol ester is a cholesterol molecule that's been esterified by the addition of a fatty acid chain. This keeps the cholesterol within this hydrophobic core as opposed to in the monolipid layer. So a chylomicron is primarily composed of triglycerides. These are from the diet, and this is the largest component of a chylomicron. Chylomicron particles are the largest of our lipoprotein molecules, and we'll see this when we compare it to both VLDL and HDL particles. There's a low percentage of protein, and these will be our apoproteins, and a small component of cholesterol from the diet and cholesterol ester, as well as phospholipids. So if we look at the packaging of a chylomicron, if we start back at our brush border with our dietary lipids dissolved into micelles, where they diffuse into the cell and are repackaged into triglycerides into the smooth endoplasmic reticulum. These triglycerides enter the Golgi complex and interface with our apoproteins, which were synthesized on the rough endoplasmic reticulum. It's within the Golgi that the combination of triglycerides, our cholesterol esters, and our proteins and phospholipids occurs to generate what's referred to as a nascent chylomicron. Part of this process initially starts in the endoplasmic reticulum with the synthesis of ApoB48. This is a protein and is being translated in the endoplasmic reticulum. From there, it's incorporated into our phospholipid particle, and our enzyme here, MTP, or microsomal triglyceride transfer protein, begins to transfer our lipids and triglycerides to form our nascent chylomicron before it enters the Golgi for maturation and secretion. This transfer protein is essential for the packaging of chylomicrons. Let's summarize this process again. Our micelles interface with our brush border epithelial cells, and the free fatty acids and monoglycerols in these micelle particles will diffuse into the cell. The bile salts in these micelles will be recycled back to the liver via the enterohepatic circulation. Within the cell, in the smooth endoplasmic reticulum, our free fatty acids and monoacylglycerols are repackaged into triglycerols. We have synthesis of our ApoB48 protein in the rough endoplasmic reticulum. And in the rough ER, we start to have the formation of our chylomicron particle that's facilitated by our microsomal triglyceride transfer protein. This starts to form our phospholipid particle with triglycerides and cholesterols. And that particle is trafficked to the Golgi complex, where it matures to form a nascent chylomicron. This nascent chylomicron has all of our triglycerols, phospholipids, cholesterol, and cholesterol esters, as well as ApoB48. These particles are secreted into the chyme and carried by the lymph. They'll enter the blood circulation through the thoracic duct. The maturation of our nascent chylomicrons is shown here. where our nascent chylomicron, associated with ApoB48, interacts with an HDL particle, which you notice is smaller, and acquires both ApoE and ApoC2. These are transferred onto our chylomicron to generate a mature chylomicron. This is essential for the function of the chylomicron. We'll talk about HDL in the next module when we talk about cholesterol transport. Just to refresh your memory from many moons ago, the apoprotein genes are translated into different proteins using the process of mRNA editing. Remember, this is where a single nucleotide is changed to introduce a stop codon. In this case, in the liver, we have synthesis of ApoB100, where the full transcript is translated, and in the intestine, where we require ApoB48 for chylomicron packaging, the introduction of a stop codon through the modification of a single nucleotide truncates the protein. So again, our nascent chylomicron with ApoB48 requires the addition of ApoC2 and ApoE for it to become a mature chylomicron. And this happens again within the bloodstream. Now let's look at the interaction of the chylomicrons with the capillaries as they begin their circulation and facilitate the storage of fatty acids. So if we start back at our intestinal epithelium, where our chylomicrons are, or nascent chylomicrons are produced, they're secreted into the lymph and enter the bloodstream through the thoracic duct. Once in the bloodstream, they acquire both ApoC2 and ApoE on the chylomicron. ApoC2 is required for interaction with the protein lipoprotein lipase. Lipoprotein lipase hydrolyzes the triglycerides into free fatty acids and glycerol. This allows for the free fatty acids to be taken up by skeletal muscle and used in a process of beta oxidation to generate energy, or the free fatty acids can be taken up by the adipose and repackaged as triglycerides as a fuel store. The chylomicron, once it's delivered its triglyceride, is referred to as a chylomicron remnant. This will be taken up by the liver via an interaction with the ApoE receptor. Once taken up by the liver, the endocytosed vesicle will be digested in the lysosomes, and you'll get a release of any remaining products such as free fatty acids, cholesterol, the proteins, and glycerol from the triglycerides. These will all be or can be metabolized by the liver through different routes. So, in chylomicron assembly, which apoprotein is transferred from HDL? The correct answer here is ApoC2. ApoC2 is required for the interaction of chylomicrons with lipoprotein lipase. This is essential for the movement of triglycerides from the chylomicron into the adipose to be stored. Remember, ApoB48 is the apoprotein that designates a particle a chylomicron. So now let's ask an important question. How do insulin levels impact the transport of dietary triglycerides? So after you eat a meal, it usually contains both carbohydrates and fats. After a meal, we know that insulin levels are elevated. So does insulin impact the uptake of triglycerides. Insulin impacts this process through the activity of lipoprotein lipase. Insulin enhances lipoprotein lipase activity, and in doing so, increases the rate of storage of triglycerides in the adipose and uptake of free fatty acids into the muscle for oxidation. So in summary, that was a brief overview of lipid digestion and chylomicron and lipid transport. You should be comfortable with the life cycle of a chylomicron and the different activities of the apoproteins involved on the particle. This is the first of several lipoproteins we'll look at. And remember, this is the lipoprotein involved in the transport of dietary fats. That will become important, an important point when we look at both VLDL and LDL particles. For your next steps, you should review the end of chapter questions. Feel free to retake the module pre-quiz and move on to module 11, session 2.